Hello, good morning, everybody. Today we are going to talk on the nervous system. And the basic unit of the nervous system is the cell neuron or the soma and its process axon and the dendrites. So, if we understand the basic unit of the cell or the neuron, we can understand how the nervous system functions, and that is the physiology of the nervous system. So all the impulses, reception and transmission, and so on and so forth, depends on the basic knowledge of physiology of the nerve fiber, nerve neuron. So my today's lecture is fully dedicated to the neurophysiology. Okay, so now we go on to our introduction part, and that is the discussion for today is the structure and the function of the neuron and the types of the nerve fiber and the types of the neurons. So these two together, the types of the nerve fiber and the types of the neuron, is the basic content of the today's topic, and that is the functions and the structure of the neuron. So to begin with, as you know, for any surgical topic or any physiological topic, you must know the structure. So structure is now evident on your display, and the picture displayed over here is a typical neuron. All the neurons are same as any cell of the body, but they have got a specialized function and they have got a special structure like axon and dendrites. So in this picture, you are seeing the cell body which has got a nucleus, it has got an endoplasmic reticulum, it has got a Golgi apparatus, and from its lower part, from one of the its area which is a thickened cell membrane, and that is the axon hillock, you have got axon and axon going and terminating with the synaptic terminals, which are also known as the synaptic knobs, and the branches of the axon, they are called the telodendria. And from the other side of the soma or the neuron, you have got a dendrites, and dendrites again has got the dendritic branches, and they are the receiving ends of the neuron, while the axon has got the transmitting function. And this is how the neuron or typical neuron looks like. Now, this is the one part of the neuron, and there is a cell body or the soma. So what it has got? It has got a roughly endoplasmic reticulum. It has got a smooth endoplasmic reticulum. The difference being the presence of the granules or the RNAs over the reticulum. And then you have got cytoplasm inside the cell body, and you have got a Golgi apparatus within it, and this is all the cell body is surrounded by a cell membrane. Within the cell membrane, you have got a cytoplasm, as I discussed, and then you have got a mitochondria like any other cell. Then you have got lysosomes like any other cell, and from the cell membrane from outside, there are dendritic spines, which are the outgrowths from the dendrites, and dendrites, they are the outgrowths from the soma. And then you have got one important structure, and that is the axon, which is coming from the axon hillock. And this is the axon hillock, and which is the showing the myelin sheath, and a small place from the base of the axon, and for few millimeters there is a lack of the myelin sheath, but then onwards each axon is covered with the myelin sheath, which is a RNA protein complex, and this is how the cell body looks like. Then within the cell you have got the basal granules. Basal granules, they are basically the basophilic bodies because they get stained positively when you are putting them on a dye, and so they are called the basophilic bodies. They stain positively with it. Then you have got, in contrast to eosinophilic bodies, which are not present in the nervous system but elsewhere. And these nasal granules, they are not actually the granules, but they are the closed cavities. And these closed cavities are having the membranes. They are the membranous cavities. And these membranous cavities are covered over the surface by the ribose nucleoprotein granules on over it. And this ribose nucleoprotein is the RNA protein complex. So these are nasal granule which are contained within the cell body. Then you have got a neurofibrillae. These are very fine thread-like structures, and they form the mesh-like structure within the cell body, and their size is 6 to 10 nanometers in diameter. And this is about the neurofibrils. Then we talk of the dendrites, which are the out extensions from the cell body, and they are usually 5 to 7 in number per cell. This is an average figure which we are giving, and they contain all the structures same as the cell body, like mitochondria, the Golgi apparatus, the nasal granules, the RNA protein complex. Everything what the cell has is being present within the dendrites, and they are the transmitting the impulses. They are receiving the impulses and then transmitting the impulses to the neuron inside, and then the process is done, and the further impulse can take place within the axon. So this is about the dendrites. Then, in contrast to dendrites, you have got an axon, which is coming from the cell body from the hillock, and hillock is nothing. It is just a thickened part of the soma, or the cell body of the cell membrane. They are the thickened parts, and from there extends the fiber-like structure called the axon. And this axon, within its cavity or from within its tube, contains the cytoplasmic. It contains cytoplasm, but they lack in the mitochondria, the Golgi apparatus, ribosome, and so on, which is contained within the dendrites but not in the axon. And one of the question in multiple choice is the difference between the axon and dendrites in terms of the contents is like this. So you must know that there is a difference in the contents of the exoplasm and the cytoplasm of the dendrites. Then this axon has got a covering with the myelin sheath, which is nothing but the RNA protein complex, and they are in sheaths. So multiple sheaths, layers piled up one over the other, forms the myelin sheath. They are the insulating bodies. They do not disturb the ingoing impulse through the axon to get transmitted to the other adjoining axons. So there is no fusing or the disturbance of the impulse which is traveling through the axon. And lastly, the axon, they terminate in the form of the synaptic knobs, and this myelin sheath, they have got a constriction after every 1 millimeter gap. and that is called the nodes of Ranvier. We will discuss this in detail, a big detail afterwards, and the myelin sheath. And so this is the myelin sheath, which is nothing but a protein lipid complex, which is covering the axon, and they are consisting of the many layers of the unit membrane, and they envelop the axon except its ending, that is the axon hillock, and at the terminal. At both the places, they are lacking. Myelin sheath is not there. And at the node of Ranvier, there are constriction in the myelin sheath at every 1 millimeter apart. Okay, and those are the constrictions are called the node of Ranvier. Then each axon at the end divides into many terminal branches, and these the terminal branches at the distal end or at their distal most end is turning into a knob-like structure, which is called the synaptic knob or the knob, and this contains lot of vesicles within it, and those vesicles are called the synaptic vesicles, and this synaptic vesicles contains neurotransmitter, and they are responsible for making the impulse to continue, and this is how the dendrites, this is how the axon ends in the synaptic knob, and they attach or they communicate their impulses through the neurotransmitter molecule through the synaptic gap and the synapses to the dendritic part of the other neuron. So if, for example, the first one is the primary neuron, then you have got a secondary neuron, and onwards it will be the tertiary neuron, and this is how the impulse pass on from one area or from one area of the neural system to the periphery if it is a efferent impulse. Okay, so this is how the functioning of a nerve in short. Then the nerve fibers. The nerve fiber, there are three different varieties: the afferent fibers, the efferent fibers, and the mixed fiber. So the afferent fibers are the fibers of the neurons which are transmitting the impulses from the periphery to the center. Okay, they may be from periphery to the center, and that is afferent. Then you have got efferent. Efferent is the fibers which are taking the impulse from the center to the periphery. Okay, so A is the periphery to the center, and E is from center to the periphery. Then you have got certain fibers which are mixed in action. They are transmitting from center to the periphery, and they are also transmitting from periphery to the center. And so depending on the traffic, which way they are going transmitting the impulse, they are termed either afferent nerve fiber or efferent nerve fiber or the mixed nerve fiber. Then you have got a types of neuron. So types of neuron, they are divided on the basis of the dendrites, maybe unipolar, maybe bipolar, multipolar, and certain they are pseudomonopolar. Depending on the number of the dendrites, they are giving extensions from the cell body. They are divided into four categories. And this is what we have learned today regarding the structure of the basic unit of the nervous system. And now onwards, we will be talking of the how the impulse is transmitted. And so all the neurotransmitter or the chemical and the extent potential and all those things we'll be discussing in our next discussion, and that will the physiological functioning of the neuron. Thank you very much, and we'll continue tomorrow with the same topic. Okay, bye bye.